This is a video tutorial for curtainptm, the post-translational modification data analysis tool brought to you by researchers from the MRC Protein Phosphorylation and Ubiquitination Unit at the University of Dundee. This project is supported by the Aligning Science Across Parkinson's Research Network. Hello and welcome to this first tutorial on curtainptm, which will show you how to upload your data. This tutorial assumes some familiarity with the curtainptm.proteo.info web tool, in particular the data upload process. So if you have not used Curtain before, we recommend watching the How to Upload Your Data into Curtain tutorial for background. So when you go onto the curtainptm.proteo.info webpage, you will see that the interface is very similar to Curtain. As with Curtain, you are required to upload a differential analysis file and a raw file. And you will notice that there are a few more drop-down menus because curtainptm requires you to include and specify a few additional columns in your differential analysis file. So let's have a look at this file. So this is a modified search output from maxquantptm, which is the software that curtainptm is optimized to use outputs from. It is possible to process outputs from MS Fragger using a custom-made script we have developed, details of which can be found in the Curtain publication. So some post-processing of the file has been performed in Perseus, the assignment of unique identifiers for each differentially modified peptide, for example, and the differential analysis. So looking at the file more closely, you can see that it has many of the same columns as a typical proteomics differential analysis comparison. But you can also see a few more PTM-specific columns, which I'll explain as I go through the drop-downs. So let's upload the files and go through each of the drop-downs one by one. So I'll just select the files to upload. Now the raw file. Okay, so now these have both been uploaded, and you'll notice that some of the drop downs have been automatically filled in because this is something that curtainptm is optimized to do. But nevertheless, even if they've been automatically filled in, I'll explain what they are. So for this first drop down, select primary IDs. So you'll remember that in Curtain we use the UniProt IDs for this column, but now that we are looking at post-translational modifications, we need a unique identifier for both the protein and the PTM. So this is why we use this unique identifier, which I'll show you in the file, this here, which has been assigned in Perseus. So for the next drop down, select UniProt accession IDs. So this now is where we put in the UniProt IDs. So this is this column here called protein. For select fold change, we select the difference column, which just has the log2 transformed fold changes between conditions. So we'll just select difference for this. For select probability score, so this is the localization probability column here, which is the probability of the detected PTM existing in the location specified by these other columns. So I'll just select this. Localization probability. So for select significant, this is the negative log transformed p-values, or just p-values because you can perform the negative log transformation. So I'll just select negative log p for that. So that's this column here in the file. Sequence window is this column here. So this is the amino acid sequence window in which the PTM is found. We'll just choose that. For this drop-down, peptide sequences, so you can see phospho STY probabilities has been selected already. That's this column here. So these are the peptide sequences where the PTM is found. with the probability of that PTM, so in this case phosphorylation, existing at that particular site. So that's already been selected. And then for PTM positions, that's a column called position. So this is the position within the amino acid sequence of the protein that the PTM is located. PTM position in peptide, that's automatically been selected as well. So that is the position then within the peptide sequence that the PTM is located. And then comparison, as I've explained before in the curtain tutorial, so this is a functionality you can use but you don't have to, but where you perhaps have multiple differential abundance comparisons in your file, and you've assigned a different identifier to each of those different comparisons, you can use this, but you don't have to select anything for this. And in my file there is a comparison column somewhere, yes, here, but it's the only number in it is 1. But of course if I had a different comparison, then I might assign 2 to like that particular series of rows, and then that would be how you could then toggle between your different comparisons within curtain. But I'm just leaving it the default selected comparison. And then for the raw file, so select primary IDs, again you want it to match with the differential analysis file, so we'll choose unique identifier again from the raw file, so that's this file here, so I've got this unique identifier column, and then I just need to choose the actual sample IDs. So I'll just scroll down, so not the log2 transformed ones, you want the data where it hasn't been transformed or imputed. There we are. And now with everything selected, we are ready to click submit. So we will click submit and just wait. This will probably take longer than for normal curtain, just because the file is that bit more complex. So now that it says finished, we can scroll down and see that the volcano plot has been plotted. And as with regular curtain, it is best to save the session at this stage by going to session and save session. And this will generate a link here like you see at the top, and also a QR code. So I hope you found this tutorial useful, and please see the next tutorial for a tour through the features of curtain PTM.